Um, so it's a pleasure to speak to you today. I don't usually speak about a disease I happen to have, so that's interesting. This is my test from this morning. I was hoping, nice solid double line, and so but I'm feeling fine. Anyways, I feel so let me just jump right into it. These are my disclosures, and I'll put the slides and references online later. But I do want to talk about the possibility of using neuromodulation. It's all in all its different forms for COVID-related disorders. I don't need to give too much of an introduction, I'm assuming, to this group about what neuromodulation is, but it's different technologies that apply directed energy into the body. Usually the name of the technology tells you how it gets the energy in. So deep brain stimulation delivers electricity deep into the brain. And by the way, these are all technologies that are broadly used within the Mount Sinai system. Spinal cord stimulation delivers electricity to the spinal cord. Transcranial magnetic stimulation, which people like Dr. Cortez and others have studied for a long time, uses induction, electroconvulsive therapy, ECT. And then you have these other technologies that are transcranial electrical stimulation, but are low power, or are non-invasive electrical stimulation directed to central structures and are low power. And so I can kind of divide these up into sort of implants, things that certainly will require surgery, interventions that certainly in their current form involving going to a medical center, but these other forms of technology, which are in principle portable. They could be used at home. They could be easily moved around in ICU. And these kind of technologies are taking on additional importance in the era of COVID. And so I'm going to be focusing a lot of these. These tend to be low risk, so risk that we will routinely use them in my lab on healthy college students. So right away, you can think about the risk benefit proposition if it's something you're willing to just stimulate young people with. They do tend to be reduced cost, and they tend to be which always matters. It certainly matters in places unlike the US, where cost is a primary factor limiting care. And as I mentioned before, they're very deployable, both for home-based and for in-hospital use. And I'll be talking about their emerging applications for COVID-19. This is something I and other people have been thinking about since the earliest days of the pandemic. This is one of many consensus projection statements written by different consortium. You can see here there's a heavy involvement from investigators in Brazil, which, like the US or many other places, has been hit particularly hard by the pandemic. And it outlines a vision for how you might use non-invasive neuromodulation. So those would be like the second two categories I just mentioned for disorders related to COVID-19. And the consensus, well, there's a couple points. The first point is that, when you're talking about targeted interventions, you sort of have to be thinking about the mechanisms by which, this virus is causing all these different disorders, the different systems that it might be attacking. And so when you're thinking about developing interventions, you're obviously thinking about interactions with systems that might be disrupted by the virus. And there's a lot of information here. And the bottom line is, I've always been interested in the effects of the virus on the nervous system. And it's a topic that I honestly feel like the more I've researched into it, the less I know. Everything I thought was for sure ends up being not for sure. But it certainly affects brain function. There's absolutely no doubt about that. There seems to be very strong evidence that it affects vascular function, and this includes in the brain itself. Whether or not it's technically neuroinvasive, I believe, is still being debated. Some people would say it's obvious. Why are you losing a sense of smell? And some people would push back on that. And so we're in so yes, you want to be mechanism based in developing intervention, but you're doing that in the context of you're not necessarily sure how the disease itself has progressed. But there are ideas. For example, you can stimulate the vagus nerve, and the vagus nerve is known to exert very powerful effects on the body's inflammatory and immune responses. So that would be a rational target. And there's other targets as well that I would talk about. So again, the first piece of it is we really have to be thinking about both how the neuromodulation technologies work and how the disease itself progresses. Related to that is the fact that non-invasive neuromodulation is not one thing. And this is something that kind of needs to be said because it's often muddled. So just like all drugs are not one thing, and you wouldn't write a consensus paper saying, do drug work for migraine? It would simply make no sense. We need to be also very careful about not lumping neuromodulation or even non-invasive neuromodulation into one thing. These are distinct techniques that you can see here. Direct the energy into the body using devices positioned on or electrodes positioned on different parts of the head or coils, and their effects on the body are potentially as distinct as different drugs. And so this is an opportunity, of course, because now you have a array of technologies that you might select from, targeting different central cortical structures, as I mentioned, the vagus nerve, the auricular branch of the vagus nerve, and so on. And I'll be giving again more examples, but certainly when you're thinking about developing an intervention for a COVID-19 related disorder, you need to be thinking about which neuromodulation technique is going to be the most effective. And in this review, we kind of thought about 4 categories of potential things you would intervene in. The first 2 relate really to the acute stages and some of the most serious concerns related to the disease. A is really trying to mitigate this pathological anti-inflammatory response. And using neuromodulation, for example, of the vagus nerve to try to mitigate that. The second category, which still is in the acute stage, is trying to assist people who are in severe distress, but treating mechanisms that are not necessarily related to the anti-inflammatory response. Then in the post-acute stage, this review categorized 2 categories. One was sort of C is just the fact that COVID kind of ruined everything. And so everything about life was disrupted, and everything that was hard became harder. And so you have more depression, more anxiety, and this obviously could occur in people who may not have been necessarily exposed to the virus. These could be societal factors. But again, this is an opportunity for neuromodulation to make a difference. And then in many cases, you have people who have difficulty making a full recovery from an infection. And so now you're looking at rehabilitation or long-term rehabilitation. And here again, you can deploy neuromodulation. And what's interesting is that in each one of these categories, there's already a decade or decades of neuromodulation being used for these kinds of applications in non-COVID populations. So it's not like out of the blue. We already have it's either FDA cleared or investigational interventions that we could already take out of the toolbox, and now we're just trying to apply them for these COVID populations. And at the time we developed this review, I think there was this other category that we weren't quite aware of at this time, but now has become very serious. It's been called long COVID. It's been called neuro COVID. Some people really don't like calling it neuro COVID. The National Institutes of Health now call it PASC. But this would be individuals who months after an infection, whether or not they were symptomatic at the time or not, are developing persistent symptoms. And I'm going to talk about this as well. And I think there's many people in the audience who have much more intimate interaction with patients suffering from these than I do. And so as I mentioned, neuromodulation already, the interest is to use neuromodulation to treat these symptoms. But again, it's not out of the blue. It's based on, for example, decades of work on using neuromodulation to treat anxiety, or using neuromodulation to enhance the effects of rehabilitation. And so again, it's being applied to these populations. Okay. During the worst day of the pandemic, when there was like police tape going up around playgrounds, and I'm quite aware that, at Mount Sinai, you were up to this to your necks. There's a lot of questions about, well, one of the questions that came up was related to whether research should even continue, right? Can we justify should we just shut down the labs altogether for an unknown period of time? And if you were to continue research, how would we continue it? At the same time, there was concern about the maintenance of clinical services. So for example, TMS is provided as an on-site clinical service. ECT is an on-site clinical service. And there was massive disruption or concern about the ability of patients to access, in these cases, these life-saving services. And so we wrote guidances for how these services could continue in that time. I mean, now I guess, 2 years looking back, it's obvious and we have our way forward, and we're not shutting down hospitals and stuff like that. But at the time, there were many questions, and so we came up with rubrics and rules of how you might manage risk benefit. It was a very biased group. We also thought that it was imperative for these trials to continue. This review also quantified the disruption in ongoing neuromodulation trials for non-COVID uses. And it was in many cases devastating, except for the exception of home-based trials. So centers that had set up home-based neuromodulation efforts, because certain forms of neuromodulation can go home, largely could continue with incremental modifications. And those incremental modifications, which I'll talk about more, including moving to 100% contactless. So as perhaps maybe before, there would be 1 or 2 on-site visits before the patient was sent home with the technology. Now it's consent is remote, all diagnosis is remote, and the device is sent to them in a box that they unpack, and they receive instructions remotely. And so the entire trial is run remotely, and then when it's done, they put everything back in the box and send it back. And I think the pandemic accelerated the development of these 100% contactless trials, and now 100% contactless interventions. So I talked generally. I want to give a few examples in these different categories. One is related to this sort of psychiatric distress. One thing that was very unusual in my experience was very early on in the pandemic, the FDA started to reach out to different investigators and different companies. And the reason this is unusual is usually the FDA is very responsive. Companies reach out to the FDA and then the FDA responds to them. But the FDA was starting to reach out to a variety of different investigators and different companies, asking them proactively, what can you immediately get running in the next few weeks? And what the FDA was concerned about was the inability of patients to access in-clinic RTMS or ECT services. And this was a real concern at the time. And so the FDA wanted there to be a strong push towards accelerating the availability of home-based neuromodulation treatments. And this led to several trials that are in some cases still running. These would be these 100% contactless trials. In many cases, they are intent to treat. That means that there's no control arm. In some cases, they are controlled studies. We're working a lot with NYU, which already had a large telemedicine-based tDCS service, and they transitioned that to be entirely contactless. Part of that as well, there's quite a bit of telemedicine. Obviously, certain diagnostic measures have to be done with telemedicine, but there's also been a lot of automation of software to collect different information and to also automate the delivery itself. So in some cases, you could have a piece of software where the intervention is pre-scheduled, 1 session per day, 5 days per week, after which they must complete the following 6 self-report questionnaires. All that is now being automated in the software. And so there is a lot of flexibility on how much manual or human interaction there needs to be, depending on how these trials are running. And these are now have been extended to long COVID. There's several trials ongoing, and results have been sort of trickling out. This is just a case report from the same group at NYU. These were 2 patients. As you know very well, with long COVID, there's a very wide spectrum of symptoms. They often include things like cognitive impairment, or what patients will describe as brain fog. And that was certainly the case in these subjects as well, but you can see they're also reporting, one is reporting numbness. Actually, both in this case are reporting numbness and pain. So it's a very diverse group of patients who are showing up. These are just case reports, but they did receive tDCS. In the case of NYU, their remote tDCS program is now a clinical service. So these patients may have actually been receiving this as part of that service, not as necessarily as part of a trial. And I don't want to, case reports are just case reports, but there is emergent anecdotal evidence of strong efficacy. But at the same time, it's also become recognized that these patients are not monolithic, they're very divergent. It's very hard to understand how, well, it's not hard. I mean, it's necessary to understand. I talked about different forms of neuromodulation, who should be getting which kind of neuromodulation, what other therapies should it be matched with, and also the difficulty of not having biomarkers to either gauge, target engagement or to gauge responsiveness. And I'll talk about these because they are in development. This is an example of another trial that just recently completed. This was run out of the Medical University of South Carolina, and this was using transcutaneous auricular vagus nerve stimulation. So this is ear clips that go in the ear, and there's a branch of the vagus nerve that innervates the ear. And so applying electrical stimulation to the ear activates this branch of the vagus nerve. And so it is a very convenient, wearable way to activate the vagus nerve with the notion of sort of engaging all of its potentially beneficial pathways, like mitigating these inflammatory responses. Now, this is not a patient. This is one of the people who ran the trial, but they're modeling the system. You can see there's actually quite a bit going on. The device that looks like an old phone that's attached to her arm, that's the actual stimulator itself. And you can see there's wires running to her ear. But in addition to that, all these patients were equipped with a home-based physiological monitoring system, oximetry, heart rate, and other measures as well. And they were equipped with a monitor that allowed them to have a televisit during stimulation. And so what was happening in this case was it was a higher touch intervention, where every time they were getting stimulation, these physiological measures were being transmitted in real time to the medical center and to the operators. And there was also a televisit at the same time. So there was quite a bit of technology involved here. And again, those results are now being prepared for publication. And it is a little bit complicated because, again, the patient started from the inclusion criteria was basically like any new neurological or psychiatric condition that did not exist previously, right? Plus some sort of positive evidence that they were actually infected. So it's incredibly broad inclusion criteria, right? You could have someone showing up and they had a headache. You could show up showing up and they have numbness of their leg or something like that. It's an incredibly broad and diverse group of subjects. So they're starting out with very different symptom burden, and so you're tracking that in a very complicated way. And so, and again, this is my test from today, but this is just a pause slide. I've been working with many medical centers on using neuromodulation for COVID and long COVID, and there's a lot of different hypotheses. And at first, I was really worried because, we don't know everything about neuromodulation. And so we have an intervention with a lot of unknowns. And then we have a disease, in this case, with a lot of unknowns. And it seems like it's kind of two unknowns. It's like, or even worse, right? You don't fully understand, and it's very hard to characterize this disease in some cases. And then you have neuromodulation techniques that in some cases are themselves investigational, or where we don't have a full understanding of the mechanism. And so this seemed like a complicated, high-risk marriage. While I think that's still true, I think at the same time, I've also come to realize that there's a tremendous opportunity here. And so I'll give one example, or I could give a few, but there's one example. There's a group in Brazil, in Paraíba, and they started to run trials for long COVID using a form of tDCS called high definition tDCS. And they were including patients who were reporting respiratory distress, trouble breathing, they were getting tired. They started to run the trial, and obviously when they first started to run the trial, they were doing physical assessments of breathing capacity. So whatever it is, it's breathing volume or breathing velocity. So they were assessing them at inclusion. What they found was, they're starting to run the trial, but when they looked back at the data, they realized that some of these patients, while they were reporting fatigue or reporting difficulty doing everyday tasks, this was not apparent in these physical measures, meaning there was no evidence that there was a dysfunction, a physical dysfunction, as far as their ability to breathe or in their lungs. Well, there were other patients who did actually have a physical dysfunction. And so what they did is they treated this initial effort as a pilot, and they reset the entire trial. And now they ran now they're running two separate trials. The first trial is with patients who actually have a measurable physical impairment, right, in their ability to breathe. The second is where they do not, and yet they are reporting fatigue, right? They're reporting symptoms that are not associated with this kind of physical, and they're running them separately. They're giving each group different forms of neuromodulation. In one case, they're directing it towards the diaphragmatic motor cortex. In their case, it's general motor cortex. They're also giving them different forms of rehabilitation for the different kind of groups, and they are reporting positive results. At the same time, there is a separate trial. This is also running in Brazil and in São Paulo with Andre Brunoni. In this case, the inclusion criteria are older patients who have experienced severe cognitive decline. And again, these are long COVID, older patients, which have are demonstrating substantial deficits in cognitive function. They're being given a different form of neuromodulation, and that's being combined with cognitive training from Brain HQ. So now you can see that actually within this problem, with this this very widespread but extremely diverse group of patients, there is the possibility to be innovative. There is the possibility to be responsive and nimble as results come to emerge, and to optimize what we're doing. And on top of that, many of these efforts are now starting to add biomarkers, including heart rate variability. So again, in Harvard, and there's a lot of interest in heart rate variability. And so I'm just saying that in this difficult and complex mess, right, there are these emerging strands now of insight, which come from, I think clinical researchers being very nimble and effective and rational in their design. Okay, I'm going to mention one more example in the context of be sensitive to time, in the context of COVID, and this was really treating the acute COVID and looking at musculoskeletal distress. This trial was just completed and published in Brain Stimulation a few weeks ago. It was run also in Paraíba, the same center I mentioned that was running some long COVID work. This was a special ICU that had been set up at the time just to support COVID-19 patients. And the inclusion criteria for this trial included being on a ventilator. These were extremely ill patients. The primary outcome of this trial was reducing number of days on a ventilator, right? And so there are standard measures to do that. This was a very complicated trial to get running and a very complicated trial to run for many reasons. The patients received transcranial direct current stimulation of the high definition version, again, directed to the diaphragmatic motor cortex. And this was combined with rehabilitation. It's always the case that I think that the most effective forms of neuromodulation are adjunct to other forms of rehabilitation. And that was done here as well. And again, I can refer you to the actual paper, but as far as the main outcome goes, and I wish I had the updated version of the slide, which shows the individual subject performance, but there was a large and significant reduction in the number of days needed for mechanical ventilation. And then with that, there was a whole bunch of other secondary outcomes like organ failure, delirium, total time in hospital, because as you could expect, if you can take someone from a hypothetical, 20 days on a ventilator to 1, their long-term prognosis is massively different. So all that was and this says N equals 19. This actually ended up being a more like a 60-person trial, I should say. And again, this was just published in Brain Stimulation, and I was very enthusiastic for it because I'm not aware of any kind of other intervention that would produce such striking effects in such severely ill patients. All right, with the time I have left, I kind of want to change gears a little bit and talk about something I call neurovascular modulation, which ends up being actually very related to interventions for COVID as it works out. The story here starts with just the notion of neurovascular coupling in the brain and the fact that it's not just neurons in our head, we have other cell types, and we have a massive density of capillaries. The average intercapillary density is something like 50 microns. So it's like capillary, neuron, glia, capillary. I mean, that's the kind of density that we're talking about. Many people understand that there is a very tight coupling between neuronal function and vascular function. For example, imaging using methods like fMRI, these are hemodynamic measures. And so what's happening is that a brain region is becoming more active and the metabolic demands of the neurons in that area increase. And then as a result of that, they call for more blood supply and blood-brain barrier transport. The blood-brain barrier is the endothelial cells that are between the blood and the brain. That function is also changed. And so it is a two-way interaction in the sense that the neurons demand different, more blood supply or more permeability, and the blood supply responds, and in exchange delivers more nutrients or is clearing more products of metabolism. So all this is very well known, and it's leveraged in many applications. And by the way, it's also known that when there's dysfunction of brain vascular, that's been linked to many neurological disorders. So the idea with neurovascular modulation is that it's not the first point. It's not, well, yes, it's the first point in the sense that when we think about neuromodulation, we cannot ignore the vascular function, right? When we're using neuromodulation to treat neurological or psychiatric disorders, we must be modulating both neurons and vascular function because the two are intricately linked. And maybe we shouldn't be treating vascular response as just an epiphenomenon, but maybe that's part of understanding the therapeutic pathway. But the second idea is that maybe we can electrically stimulate this, the vascular supply itself, or for example, specifically the endothelial cells that form the blood-brain barrier, so that they are now the target of stimulation. And this reverses what the normal direction of what people think about for neurovascular coupling, because now the vascular goes first. There's more blood supply, there's changes in transport across the blood-brain barrier. That's what the electrical stimulation does. And then that leads to neuronal effects. And of course, nothing's ever going to be so isolated, but that is the idea behind neurovascular modulation. And we've been looking at it for deep brain stimulation. We've been thinking about it for spinal cord stimulation. And we've been thinking about it for forms of non-invasive electrical stimulation, including transcranial direct current stimulation, which was a technique you saw was used in a lot of these COVID trials. And I should say up front, no one's saying that electrical stimulation doesn't affect neurons, right? That doesn't make any sense. In fact, in my own lab, I've spent decades characterizing the effects of electrical stimulation on neurons in brain slices. So there's no vascular, it's a great system. There's no vascular supply in that system. However, in the intact brain, the blood supplies are obviously always there. And as I mentioned before, in tDCS, as is in other forms of neuromodulation, everyone knows that there's a vascular response to stimulation, but it's considered an epiphenomenon. It's like considered not interesting. It is considered, or it's interesting only in the way that it helps us image neuronal function. But other than that, I mean, you apply stimulation, whether it's tDCS or other forms of stimulation, you see a profound vascular response. Thank you. That means the neurons are responding, and you kind of toss away the vascular response itself. But again, the proposal here is that if you took the neurons out and left only the vascular response, right, only the blood vessels itself, would they still respond to stimulation? And if that did, it actually opens up the door to many interesting therapeutic pathways and mechanisms of treatment that I can talk about. And again, I need to say again, no one is saying that electrical stimulation doesn't affect neurons. It's something that I and other people have been working on for a long time. So we're talking about an adjunct mechanism, right? We're talking about something else here. It is very hard to prove this. And the reason is that if I were to apply electrical stimulation to anybody's head and show a response, people would assume, well, that's because you're stimulating the neurons. And it's sort of like the de facto assumption. And it's very hard. And then when you see the blood supply respond, people will just say, well, that's only because you stimulated the neurons. It is very hard to unwind this issue. So one approach we did is we essentially have models where there are no neurons. So these are in vitro models, which contain only the blood-brain barrier, or even only one cell type of the blood-brain barrier, like endothelial cells. Now, these models have been around for a long time. They've been developed because there's been a long-standing interest to study blood-brain barrier function. So these are essentially wells, and you have, in this case, you're looking at endothelial cells, and they've been grown inside these wells, and they form these tight junctions between them. So essentially, you have a barrier. You have a monolayer barrier, and there has been decades and decades of work characterizing how these blood-brain barrier models work. And there's many tools that you can use to study their response. The only difference we did is to these existing systems is we added electrical stimulation. So you have all the existing tools to measure transport and function of these blood-brain barrier model systems, only now we are zapping them and asking, how does this function change? And there's many things you can do. You can image transport across these. You can, for example, fluorescently label different molecules and image how easily they're transported across these blood-brain barrier models. You can also measure water transport across these barriers. So that's what this bubble tracker is here. So there's a slow and constant flow of water across your blood-brain barrier. And that is something that can also be quantified here as well. And that ends up being important for what I'm going to say next, because what we found is that direct current stimulation produces an acute, reversible, very deterministic change in water transport across the blood-brain barrier. That's what you're seeing here with this bubble. The bubble is moving at a certain rate, and depending on the direction of electrical stimulation that we apply, we can make the bubble move faster or slower. And what that means is that water transport across the blood-brain barrier is being either increased or decreased. And again, there's a lot of details here. I mean, this effect you're seeing here, for example, is this natural sealing of these models that happens over time. But the effect is very robust, as in trial by trial level of robustness. And not only that, it's very explainable. So there's a theory, there's a mechanism, something called electroosmosis. And electroosmosis says that when you have a tight barrier, and the blood-brain barrier is a very, very tight barrier, and you pass current through that barrier, you will drag water through it. And the amount of flow that we're seeing here is exactly consistent with what we predict based on the theory of electroosmosis, factoring things like how tight the barrier is. So what that means is that when you apply direct current stimulation to blood vessels, you drag water across them. And we sort of unpack the consequences of this in several papers. But it turns out that when you drag water, you drag things with the water, like different kinds of molecules. And it also turns out that that dragging of water and the molecules with it is a very powerful signal for the blood-brain barrier itself. It senses this flow of water and it structurally and molecularly changes as a consequence of it. And this is almost like it's almost like plasticity at the level of the blood-brain barrier. So without any neurons present, first of all, these systems are responding. They're responding in ways that we think could be very profound as far as changing neuronal function. And in some cases, like electroosmosis, the effects are immediately reversible. In some cases, they are persistent. This has been a very active area of research for myself and other people in the past few years, and I think it has some very interesting implications. First of all, if you can affect vascular function, if you can enhance directly blood flow or transport across the blood-brain barrier, then you can affect neuronal function. And this may have implications in areas such as neurorehabilitation, where you want to produce a transient increase in function of a particular brain region. And I should mention, we've extended this work not just from in vitro, but also in vivo, using techniques like multiphoton imaging, where you can image individual capillaries in the brain during electrical stimulation. Though, of course, now you have to contend with the fact that the neurons are there as well. The notion of being able to directly stimulate the blood-brain barrier and vasculature, but in very specific ways, also opens the door to very compelling mechanistic pathways. So I mentioned this water flux. So imagine if you could put a cap on someone and for 20 minutes increase water flow through their brain in such a way that actually acts to, let's say, clear the accumulation of things you do not want in the brain, right? So this is sort of this notion of flushing the brain or clearing the brain. And this is very speculative at a translational level, but we certainly see the evidence for this at the cellular level. And so the notion that you can have a technique that, again, separate from any neuron effects at all, can be affecting brain clearance processes, opens the door to many interesting therapeutic pathways. And the final area that we're very interested in is actually as far as its neuroprotective applications. So there are currently trials running, these are at UCLA, where stimulation is being applied within an hour of patients being wheeled in to the ICU after stroke. And the notion here is that, yes, there may be a region of the brain that is destroyed and unrecoverable, but there may be a penumbra around it that is suffering from reduced vascular supply. And maybe if we can transiently boost blood flow in that penumbra, this can exert an emergency protective effect. And there are emerging very striking results from these trials, which, as you can imagine, are also extremely complicated to run because these patients are extremely unstable to begin with. And there's also safety concerns as well about stimulating the vascular supply in patients with a compromised or injured vascular system. So these are really, Dr. Bar-Hussein has really been leading these efforts at UCLA, and hopefully this will be expanded to a multi-center effort, but these are very difficult trials to run. And so just to put like a pin on it, so the notion behind neurovascular modulation is that neurons are not alone in the brain, like, of course, right? And so there are other things in the brain. There are blood vessels that are formed of endothelial, there are glial cells who specifically are tightly coupled to the blood-brain barrier itself. There are, for the larger blood vessels, nerves that specifically innervate those nerves. We have many kinds of blood vessels, from these dival penetrating arterioles, the meninges itself, which is going to get a very high level of current whenever we stimulate, has a lot of blood vessels, and so on. And so you have this rich sort of target base. And so the first aspect of neurovascular modulation is even if we're directly activating neurons, maybe we cannot understand how we stimulate neurons without understanding this coupling. So we can't understand, for example, how electrical stimulation could boost plasticity, which it does, without also understanding how endothelial cells and glial cells are all coupled into that process. And the second aspect of neurovascular modulation is that we might actually be reversing the traditional recruitment order, making blood vessels activate first. And again, as I tried to indicate before, I think this has very interesting dimensions to it as well. And this was something we reviewed or tried to characterize in this recent paper as a concept. I want to just throw out one more interesting, or to me, interesting idea. My group does a lot of current flow modeling. What we like to do is we like to imagine that a neuromodulation technique is being applied to the head, and for it to work, that energy must enter the brain. And we're very interested in where that energy enters, how much of it enters, maybe only 1% of it enters, and also where it goes, which brain regions get a lot of energy and which brain regions get a little bit of energy. And so we've done a lot of work on that for tDCS, for example, where we and others have shown that when you apply tDCS using 1 milliamp of current, very small amount, you produce an intensity inside the brain that we quantify in terms of electric field as 0.4 volt per meter. So this is like the bulk electric field in a voxel of the brain. But now we know that inside that brain are capillaries, which are illustrated here in the next slide. And we know that these capillaries are extremely resistive. The blood-brain barrier is unbelievably resistive because it's designed to be very, very, very tight. The brain around it, what you call the parenchyma, is relatively conductive, and the blood is relatively conductive. So what you have here are these tubes that are incredibly resistive, and they're filled with blood, which is conductive, and they're surrounded by parenchyma, which is conductive. And what we found using computer simulations is that as a result of that, a small amount of current will cross across the blood-brain barrier, but because the blood-brain barrier is so resistive, this will lead to massive electric fields. So we predicted that, for example, in the case of transcranial direct current stimulation, though you have less than 1 volt per meter around neurons, you can have several hundreds of volts per meter in the blood-brain barrier. And again, this is almost like a inevitable consequence of these physical properties that I just mentioned. It's the fact that you have these very resistive walls surrounded by relatively conductive media. And so we've done this analysis in some detail. This is all theoretical work, but we're basically predicting that going from the parenchyma, which is what most people think about when they talk about electric fields, to the blood-brain barrier, you can have 200, 600-fold amplification, depending on what's going on. So tDCS produces 0.4 volt per meter in the parenchyma, leading to 100 volt per meter across the blood-brain barrier. But other techniques like DBS or TMS or ECT, they produce 100 volt per meter in the parenchyma, and they get the same level of amplification. So this is going to lead to 10,000 volt per meter across the blood-brain barrier, or kilovolts. This is a very controversial prediction, but that's okay. We've all made controversial predictions before, and sometimes they're right and sometimes they're wrong. But the reason I want to emphasize this is it literally reconcentrates our attention on the fact that maybe we could be stimulating across all these neuromodulation techniques the blood-brain barrier, that the blood-brain barrier might be the primary target or at least a very important target of neuromodulation. Because of that. And we're now actually coupling these electric field models to transport models to get at this notion of flushing that I mentioned before. So it turns out we can predict that as well. These are kind of pretty pictures we get. This is actually a prediction of not just the current flow, but the change in water transport produced around neurons as a result of this electroosmotic idea. So this is my last slide. I tried to get you interested in two things. One is I'm very interested in the possibility that neuromodulation could be used to address COVID-19 related disorders. And I increasingly believe that this can be done rationally. I think this could be mechanism based. I think this can advance the science of both how we understand neuromodulation and how we understand COVID and things like long COVID. But this is very hard work. And I'm also very interested in the notion that despite having worked for 10, 20 more years on the effects of neuromodulation techniques like tDCS on neurons, I'm now thinking, well, maybe it wasn't the neurons. Maybe all along we've been stimulating blood vessels. And things that we've known all along, like that all forms of neuromodulation, DBS, TMS, all that, they produce profoundly strong signals in the fMRI. Hold on, maybe that was the effect of stimulation and not just an epiphenomenon. In all these cases, of course, we can't, it's appealing to oversimplify, but we shouldn't, because these are obviously the brain, everything's coupled and complicated. But I feel like this is a very compelling area of work. And finally, I would say that these things actually could be connected in the sense that, for example, if COVID affects vascular function, or if activating vascular function could treat COVID-related disorders. So, okay, thank you for your attention.